Now, today's topic is the fertilization. Dear students, as you know that the fertilization is the fusion of male gamete with female gamete. This process was discovered first by Strasburger in 1884 in Monotropa plant. Now, the first of all, the pollen-pistil interaction. After pollination, what happens? Stigma receives various types of pollens. Pistil has the ability to recognize the right pollen. To accept or reject, there goes a continuous dialogue between the pollen and the pistil. This is called the pollen-pistil interaction. This dialogue is mediated by the chemical components of the pollen interacting with those of the pistil. If the reaction is favorable, the pollen grain germinates on the stigma to produce the pollen tube through one of the germ pores. These germ pores are present on the pollen grains as we have discussed it about in the microsporogenesis. All the events from pollen deposition on the stigma until pollen tubes enter into the ovule are together referred as the pollen-pistil interaction. Or we can also say that the pollen-pistil interaction is a dynamic process involving pollen recognition followed by the promotion or the inhibition of the pollen. Now the growth of the pollen tube. When the pollen tube comes down from the stigma into the style, first of all, the generative nucleus, this cell, it's a generative cell and its nucleus is known as the generative nucleus. It enters into the pollen tube. Then it is followed by this vegetative cell enters into the pollen tube. Then it is followed by the generative cell. So that means the vegetative nucleus enters, then the generative nucleus into the pollen tube. Now the male gametophyte, the tube nucleus always occupies the terminal position in pollen tube and controls the growth of the pollen tube here. First of all, this tube nucleus enters into this pollen tube. This is a pollen tube. Then what happens? Meanwhile, the generative cell divides mitotically to form two male gametes here. 1 and 2. These are the two male gametes. Both of the male gametes are the non-motile, that means they lack the flagellates. One important point is here that the boron and the calcium, mainly the boron, is essential for the growth of the pollen tube. For that, best temperature is 20 to 30 degrees Celsius. Pollen tube shows apical growth and chemotropic moment. Now the entry of pollen tube into the ovule. Finally, the pollen tube enters into the ovary. At that time, what happens? Ovules become mature. Inside the ovary, obturators, that is the tissues, some sort of tissue or the outgrowth of the placenta, it guide the passage of the pollen tube towards the micropyle. This is the micropyle position. A mature ovule is that in which embryo sac has also matured. In the ovule, pollen tube can enter into three places, such as this one, micropylar, that one, it is the chaliza point, or through any integument. Now the progamy, it is known as progamy when pollen tube enters into the ovule through the micropyle, it is known as progamy. It is found in most of the angiosperms. It may be called chalazogamy. When in chalazogamy, what happens? The pollen tube enters into the ovule through the chalaza here. Through the chalaza when it enters into the ovule. This method was reported in Casuarina by Tribb in 1891. The common examples are the betula, the paper tree, or the Jugulanus, the walnut. It is also known as mesogamy. In mesogamy, what happens? The pollen tube enters into the ovule either through integumentous, that is in case of cucurbitous, or through funiculus, that is in case of populus. Now the entry of pollen tube into the embryo sac. Pollen tube can enter into the ovule through any passage, but inside embryo sac, it enters only through the egg apparatus. That's the micropylar side. It's a common. Commonly it enters through the micropylar side, that is degenerating synergid cell. After the entrance inside the ovule, it grows toward the egg apparatus because synergid cells secrete the chemicals, those that are the hormones which attract the pollen tube towards the synergids. It means pollen tube shows chemotropic movement in ovary. Now the release of pollen tube contents into the synergid. Uneven synergid starts degenerating when the pollen tube comes near the egg apparatus. The pollen tube enters into the embryo sac through the degenerated synergid. When the tip of the pollen tube enters into the embryo sac, vegetative or the tube nucleus, it degenerates. The tip of the pollen tube swells and then bursts due to the endosmosis. After reaching inside the embryo sac, the pollen tube releases all contents, including both male gametes, inside the degenerated synergid of the embryo sac. Here you can observe the two dark granules appear in the region of the degenerating synergid. These are known as the X bodies. Both are degenerating tube nuclei and the degenerating synergid nucleus. Here you can see here, this is the central cell. This is the egg nucleus here. These are the plasma. This is the synergids. And here these are the filiform apparatus which we have discussed in the megasporangium. And here you can see this is the vegetative nucleus and these are the male gametes. And this is the secondary nuclei. Here these are the two male gametes which reach inside these synergids. Then here this one male nuclei, it moves towards the egg, this one, and this one moves towards the secondary nucleus. Now the fusion of gametes. After bursting, pollen tube releases two male gametes. One male gamete fuses with the egg to form diploid zygote. This fusion is called syngamy or the generative fertilization. Here the male gamete haploid, it fuses with the egg haploid. As a result of this, a diploid zygote is formed. The other male gamete fuses with secondary nucleus. That means it is a diploid structure to form primary endosperm nucleus, which becomes a triploid structure. This fusion is called a triple fertilization. That is the fusion of three nuclei. It is also known as a vegetative or the pseudo fertilization or tropomixis. Here the male gamete, the haploid, fuses with the secondary nucleus or diploid structure and results in the formation of a triploid structure, that is a PEN, primary endosperm nuclei. Now the release of male gametes and the double fertilization. This is the pollen nuclei, this is the egg nuclei. The pollen tube bursts and releases two sperms, which enter here. These are the two sperms inside this embryo sac, inside the ovule. Then what happens further? Triple fusion takes place. Here this one fuses with this polar nuclei and it results in the formation of triple fusion. That's a polar nuclei plus sperm, whereas another one it fuses with the egg, results in the syngamy, that is the egg cell plus sperm. So this is called also the double fertilization. As a result of this double fertilization, primary endosperm is formed, which is a triploid structure, and also here a diploid zygote is also formed. Remember, since two types of fusions, that is syngamy and triple fusion, takes place in an embryo sac, the phenomenon is termed as the double fertilization. This event takes place only in angiosperms. 5 nuclei are involved in double fertilization. It was discovered by Nowotny in Lilium and the Fertilaria and was later on confirmed by Gignard. Now in the coming these lectures we will discuss about the post-fertilization events. Thanks.